Good evening, doctor. Myself Gandharavali. Now I am going to explain about polymerase chain reaction. It is called polymerase because the only enzyme used in this reaction is DNA polymerase, and it is called chain because products of first reaction become substance of the following one. What is polymerase chain reaction? It is a technique generating large quantities of specified DNA, and it is a cell-free amplification technique for synthesizing multiple identical copies of DNA of interest. It is originally developed in 1983 by American biochemist Carey Mullis. Now the principles of PCR. The double-stranded DNA of interest is denatured into two separate individual strands, and these each strand is allowed to hybridize with a primer in the process of renaturation. This primer template duplex is used for the DNA synthesis by the enzyme of DNA polymerase. Denaturation, renaturation, and synthesis are repeated again and again to generate multiple forms of target DNA in the process of the polymerase chain reaction. Now the requirements for the polymerase chain reaction. A target DNA, two primers, 4-deoxyribonucleotides, a DNA polymerase that can withstand at the temperature up to the 95 degrees Celsius, and a PCR involves repeated cycles for amplification of target DNA, and the thermal cycler to regulate temperature during the cyclic programs. And each cycle has three stages: denaturation, renaturation or annealing, and synthesis. Now first let us know the stages of the PCR, and the first stage is denaturation. In this process, the temperature increases about 95 degrees Celsius for 1 minute, and the DNA now gets denaturated and forms two separate strands. Now next stage is renaturation or annealing. In this, the temperature slowly cools down about 55 degrees Celsius, and the primers base pair with complementary regions flanking target DNA strands. And the third stage is synthesis. In this, the initiation of the DNA occurs at the 3' hydroxyl end of each primer, and the primers are extended by joining bases complementary to DNA strands. The synthesis process is comparable to DNA replication of leading strand, and the optimum temperature should be maintained as required by the DNA polymerases. These are the stages of the polymerase chain reaction just now, as we discussed: denaturating stage, annealing stage, and the extending stage. Thermus aqueous DNA polymerase, its optimum temperature is around 75 degrees Celsius, and for E. coli the optimum temperature is around 37 degrees Celsius. This reaction can be stopped by raising the temperature around about 95 degrees Celsius, and each cycle of the PCR takes about 3 to 5 minutes. Now let us move to the PCR and cycles. And the cycle 1 is the new DNA strand joined to each primer beyond sequence is complemented to the secondary primer. New strands are referred as long template. They will be used in the second cycle. Cycle 2, the DNA strands are first denatured, annealed, and with primers and subjected to DNA synthesis. At the end of the second round, long templates and short templates are formed. In cycle 3, the original DNA strands along with the long and short templates are starting materials. First, denaturation, renaturation, and synthesis are repeated. This process is repeated again and again and again many times. At the end of the 30-second cycle of PCR, about millions fold target DNA is synthesized. And the sources of the DNA polymerases. Lino fragment of E. coli DNA polymerase is used in original technique. This enzyme gets denatured at high temperature. Therefore, fresh enzyme has to be added for each cycle. For thermus activius DNA polymerase, it is heat resistant, so no need to add freshly. And this enzyme Not necessary to freshly add this enzyme for each cycle. It can be used as many times maybe. Now variations of PCR: nested PCR, inverse PCR, anchored PCR, reverse transcription, asymmetric PCR, real-time quantitative PCR, random amplified polymorphic DNA, amplified fragment length polymorphism, rapid amplification of cDNA ends. Now applications of PCR. In prenatal diagnosis of inherited diseases, it is used for prenatal diagnosis of various diseases by chronic villus samples or cells from amniocentesis, like sickle cell anemia, B thalassemia, and PKU can be detected. In diagnosis of retroviral diseases, it is used for the diagnosis of HIV infection. For diagnosis of bacterial infection, it is used to detect bacterial infections like tuberculosis. In diagnosis of cancers, it is used to detect the cervical cancer. PCR in forensic medicine, and it is used for the identification of the criminals. Thank you.